Hey everyone, welcome to our next Boltzmann session on mathematical models for reproduction and evolution. Our speakers today are Saurabh and Burhan, fourth year undergraduates from IIT Madras. You can ask your questions in the comment section and we'll answer them after the talk. Burhan and Saurabh, over to you now. Burhan?
Yes, thank you for the introduction. And thank you for the opportunity to present us in this session. So today we are planning to talk about mathematical modeling of reproduction and evolution as a part of the Boltzmann lecture series. So this is the overview of the talk. Initially, we will set up the question and then look at it from an information theory perspective. And also we'll encounter Maxwell's demon and second law of thermodynamics and so on. And then we will look at some validity assumptions and what are the implications in some real life examples. So as we are aware that reproduction is an essential feature of all living organisms. So many unicellular organisms usually undergo asexual reproduction, where a cell divides into two or in multiple smaller fractions, and and all of them are identical copies of each other, nearly identical, because the DNA copying mechanism is not. It's very efficient. But it is not error proof. So there are some errors, and which lead to some variation in the population. Also, there is sexual reproduction, where there are two sexes, male and female, and their gametes fuse with each other to form the embryo, or that goes on to become an individual. And there are some species which can undergo sexual as well as asexual reproduction. Some of the examples are self-pollinating plants. Now there are slight variations among the individuals. And the population can keep growing as there would be resource constraints, and because of these space and resource constraints, there will be competition among individuals of the species, or even among multiple species, and because of that, there would be evolution, or in some sense, there would be some selection of some individuals that are better suited to that environment. They will survive and reproduce, and these species will gradually evolve. So these laws are given by Charles Darwin. Now, one of the mistakes that we often tend to do is we call it survival of the fittest. However, fittest is a very context-dependent term. So, for example, in sickle cell disease, so sickle cell disease is a genetic disease in that there is one mutation in the gene that encodes for hemoglobin. Now, because of that small nucleotide change, or there are letters of A, C, T, G, and there is some flip in one of the nucleotides, because of which the red blood cells take up a sickle cell form, and because of that, it cannot. The carrying capacity of the oxygen reduces. Now, this is a genetic disease, and these are the molecular bases of the diseases. However, so on the right-hand side, I have, on the top, I have the map of malaria prevalence in the world, and at the bottom, I have the prevalence of sickle cell disease. So you would see a startling correlation among the countries that have high sickle cell disease, and that is because Sickle cell disease also confer some sort of resistance towards malaria. And malaria is often found in tropical countries where the mosquito, its vector, can reproduce. So, so the individuals with sickle cell might seem unfit in an isolated condition, but when you add a selection pressure of let's say malaria, maybe they would thrive much better than the other ones. So again, fitness is a very context dependent and very complex term. So now our model is adopted from this book of Information Theory, Inference and Learning Algorithms by Professor David J.C. Mackey. Now I'll set up the problem. So let's say the genotype is, so genotype is basically whatever is there in the DNA. And so there are two terms, genotype and phenotype. Genotype is whatever is there in the DNA. Phenotype is whatever the manifestation of the organism is. So let's say genotype is some genes, X, Y, Z, and phenotype is, let's say, the height of the individual. So the genotype is encoded as a vector X of G bits. So G is the length of the vector X. And X is such that if the first bit is bad or unfit, then it is 0. If the next one is good or fit for the individual, then it's 1, and so on. So it is of the length G. And the way we are estimating the fitness of the individual is we are taking sum across all X of G, which is basically we count how many of them are good states or how many of them are fit. Now you can think of this vector X as the bits stored in the genome, and it could be thought of genes like each of this block can be a gene A, B, C, whether the gene is unfit or fit, or it can be nucleotides. It can be letters A, T, G, C, and if there is a flip, then it manifests something that is unfit. Okay, so now I'll hand it over to Saurabh.
Yeah, can you share my? Can you see my screen?
Yes, Saurabh.
Okay. So we'll basically look at mathematical models of two types of reproduction system. One is simple asexual reproduction, where the parent gives rise to two progeny, the parent dies, and the progeny undergoes some mutations in their genes. Like each bit in the genome sequence flips with some probability m. And the next model will be sexual reproduction, but without we will study it without mutations. Just the child will have one of the bits of the parents with some probability half, and we'll study what that happens. We have not included mutations plus sexual reproduction, although we have simulations for that available. I'll share the Jupyter notebook for that afterwards, so you could play around with that. So we'll start with theory of mutations. The theory of mutation is the same way I described it. Each parent gives rise to two progeny. The parent dies, and each of the progeny has bit flips for each bit with equal probability of m. Then afterwards, to keep the population constant, we just choose the top 50 percentile of the progeny. So in one stage, the number of organisms are doubling, and then we just take half of them, so it remains constant with time. And then those selected progeny serve as the parent for the new generation, and the process continues. So our fitness model, as Burhan described, is just the sum of the good bits present in the genome. And for convenience, we'll work with normalized fitness. So you can see in this expression for fitness, capital F can take a value between 0 and capital G, so we'll just divide it by capital G, so it stays between 0 and 1, and it's easier to interpret. One can observe that but if an individual just undergoes simple mutations, then one can easily achieve a fitness of 0.5, because suppose you have more good bits than bad bits and no selection, so and you keep on flipping those, eventually you'll land up with exactly half good bits and half bad bits. And the same thing if you have less good bits than bad bits. So we'll work with something called excess fitness, which is just small f minus 0.5, and we'll see how that evolves with time. If you use the law of iterated expectations, you can find that the mean and excess fitness and variance is given by delta f bar for the progeny is equal to 1 minus 2m times delta f. By this thing, you can also see that this is pretty clear. This is pretty intuitive, because if there are more good bits than bad bits, then since each bit flips with a probability m, then at the end you expect more good bits to flip to bad bits than bad bits to good bits. So mean will decrease in for the progeny, and variance will be just, we can just model it as a sum of Bernoulli random variables and with parameter m, and then this is a very familiar expression we have. If the parents have some excess mean fitness delta f of t, and variance is beta times m of g, taking inspiration from this form, then the selected progeny, which is you could imagine as suppose you start with the distribution for the mean fitness of the parents, and then it gives rise to two progeny and with mutation. So that distribution shifts to the left, its mean shifts to the left, and its variance also changes slightly. And then you choose the upper half of the distribution, and that serves as the parents. For the selected progeny, the mean excess fitness is given by this thing. How do you this is pretty clear. This is the mean fitness of the parent, and this is some proportionality constant times the variance of the parent, because variance measures the spread. So we expect that after selection, the mean for the selected upper half of the distribution will move to the by some amount which is proportional to the standard deviation. This is pretty intuitive, and variance for the next variance for the progeny in terms of the original one is given by this relation. We assume something called dynamic equilibrium for the variance, which is that variance for the progeny is the same as variance for parents, and it remains roughly constant with time. There need not be an equal to sign here. It can change by a factor of 1, order of 1, but not a factor of 10. And you intuitively again by that shifting of a curve to the left and then selecting the upper half of the distribution, etc. But for a more convincing argument for why this holds, I'll show you the simulations for this, and then you can probably experiment with this later. When can this fail? This fails when your mean excess fitness is close to 0 or close to half. So that's expected. If your mean fitness tends to 1, then you would expect that the variance dramatically decreases. And so we have this model, and for simplification, we just assume a Gaussian distribution for the parents, and then it shifts to the left. The upper half is selected, and so on. As alpha, beta, and gamma. Is Gaussian a good probability distribution selected? Well, Gaussian is we know that sum of independent identically distributed random variables tends to Gaussian, but yeah, we have this selection of the right half of things, so maybe an distribution will be better, but we don't really care about what these alpha, beta, and gamma are. Maybe these factors could be something different for Poissonian or Gaussian, but we just want to see the time scales involved and the steady state analysis and all that. We really care about how M and G appears rather than the prefactors for those. So if you substitute these for Gaussian distribution, alpha, beta, gamma, you get this differential equation for mean excess fitness. Sorry, there's a typo here. This should be d delta f by dt equal to minus 2m delta f plus another typo here. It should be square root of m by g. And then you can just differentiate this again, and then you'll have a linear differential equation in the derivative of excess fitness, and you just integrate it out, and you get the final excess fitness to have this form. The proportion, the constant C will be fixed by the initial condition. If you start from initial condition of 0.5, then you'll get some different constant, and we get the steady state excess fitness to be this thing. Now, how do you interpret this thing? Of course, this delta F cannot be more than half. So another reason why this assumption fails is when M into G is much smaller than 1. So one can observe that if M into G is greater than 1, then you won't ever achieve a mean fitness of close to 1. And if M into G is less than 1, then you will exactly achieve mean fitness of 1. How do you interpret this? M into G is the expected number of bit flips every generation. So you can interpret it as some kind of a gradient descent in a multidimensional function. The function is a mean fitness, and there are G variables which are the bit values, G bits, so G input values, and M into G is the fluctuation from the original value to the new value in case of the progeny. So if the fluctuations are smaller than 1, each of the input is only 0 or 1. So if the mean distance you go away from the original point in every generation is smaller than 1, then you expect the evolution of the mean fitness to eventually come down to the bottom of the value or something. But if it's greater than 1, then you expect some kind of large fluctuations which prevent it from going to the most stable point, and there will be non-zero variance as well as non-zero deviation from mean fitness equal to 1. And now I'll go to the theory of combinations. I'll show you the plots and simulations and animations for both of these after I finish this section. Now this is the theory of combination. It's pretty simple. You have a population of size P. Then you divide these P individuals into pairs randomly. Then each of the couple gives rise to 4 children, and a parent, both of the parent die. And for each children, each bit can take a value of any of the parent bits with probability half. And this is very close to what we get in real life. And this is much easier to, the differential equation for this is much easier to obtain than the theory of mutation differential equation. We assume 2 things here. One is that bits are uncorrelated. So bit 1 is uncorrelated from 2. And we also assume homogeneity, that fraction of bits that are in good state are same for all of the bits. So if Fg is the probability that the bit G is in good state, then we assume that Fg is same for all G in capital G. That is, Fg is equal to small f, where f is the normalized fitness here. So this mean can be easily found out by a sum of a combination of random variables. So that will be the same as the parent's mean, which will be f. And the standard deviation is again a sum of Bernoulli random variables. The parameter for each Bernoulli random variable is f. So adding all of these up and taking mean, we get this kind of expression for standard deviation. So what happens here is a probability distribution curve for the parent's fitness. Then the parents give rise to children, but unlike in the theory of mutation, the mean fitness for the children is the same as the mean fitness of the parent. Then you select the upper half, and the process continues. So we write this in differential equation form, that the mean shifts to the right by an amount proportional to the variance, as again eta is the proportionality constant here. This depends on whatever curve you have. Again, we use a Gaussian curve to model eta here, but it's again a proportionality constant. You can use a different curve, but we are just interested in the dependence on capital G, etc. What happens, what are the time scales and all. And this is a pretty simple differential equation which you might have studied in JE or something. You just bring this numerator to the bottom, you take dt up and you integrate, and this is the solution you get for the differential equation. C is again the constant which is determined by the initial conditions. So one can see that the mean fitness reaches the optimal value in roughly pi over square root of pi into square root of g by eta generations. Now I'll show you the curves for the evolution for both of these cases. So this curve is for, I'll just zoom it, for simulations. I have to switch that on, right? Just a minute. Yeah, I'll show the simulations for both of these cases. Yeah. So this is the fitness evaluation for asexual reproduction. Small m into capital G is the expected number of bit flips you after each generation. So if small m into capital G is greater than 1, then you can see that the normalized fitness will never go to 1. If small m into capital G is lesser than 1, then you expect it to go to 1. Of course, the time to reach 1 depends only on m and not G. So the further curve for this is not visible here, but it is plotted over here. This is for small m to capital G equal to 0.5, that is 0.5 expected bit flips per generation. And you can see that it quickly goes to 1 after some 100 or something iterations. And this is again the standard deviation evolution. Remember, we took the dynamic equilibrium case for the variance, and you can see that it roughly remains constant. Of course, there's a small dip because we started from 0.5, as I said. You expect some deviation when your mean fitness is near 0.5 or close to 1. But in between other values, it roughly remains constant. It does not change by a large factor. Order of 1 changes in factor, we are pretty okay with that. This is the fitness evolution for sexual reproduction. The blue curve represents sexual reproduction without any mutations. So this resembles a sine-like thing. You can see that it's not an exponential thing, exponential curve. It quickly goes to 1 and then stays at 1 rather than some exponential saturation or something. And when you have mutations which are not 0, you expect that the normalized fitness will not go to 1. Of course, you have not studied this model due to lack of time, but the simulations are available to play with. I shared the Jupyter notebook after. Assumption we took was we assumed homogeneity and uncorrelatedness. This happens when the population size is pretty large. So if you have say 10 populations reproducing sexually, then you expect some kind of inbreeding to give correlations and bad bits becoming frozen into the genome. For reference, see the Habsburg jaw consequence. Habsburg were the royal family of Austria, and they intermarried within each other, which gave rise to jaw defects. But again, if the population size is small, but you have non-zero mutations, you can overcome this barrier. This is what this curve shows. Now I'll just briefly show an animation for how the fitness evolves with time for sexual and asexual reproduction. This is the sexual reproduction. Also note that in comparing sexual and asexual reproduction, the time constant for sexual reproduction to reach 1 depends on root G and some eta, which is some order of 1 constant or something. But for asexual reproduction, the time constant depends on M. Sexual reproduction reaches the optimal value much faster than asexual reproduction, and that is why the dominant species on Earth are those kind of species rather than amoeba and bacteria being at the top of the food chain. This is the curve for sexual reproduction, and you can see that it moves faster than asexual reproduction. And you can also see, if you change the frame rate, I'll provide the code, that for the children, the parents give rise to children, the mean does not change. Only after you select the upper half, the mean changes. So that's the end of part of this mathematical modeling of fitness. Now Burhan will talk about implications of these, then we'll have some information theory perspective, and I'll encourage Burhan to take over.
So thank you, Saurabh, for setting up the problem. And yes, so can you see my screen?
Yes, Burhan.
Okay, thank you. Yes, so here from the previous model that Saurabh has explained, here we try to sort of find the maximum tolerable mutation rate. So now we have combined two parts of the fitness function. So let's assume that an organism can undergo sexual as well as asexual reproduction, and the fitness rate, I mean, we are taking a continuous approximation, and with respect to time, it would change in this particular order. So the first term is from the asexual, mutations because of asexual reproduction, and the second term is mutation because of sexual reproduction. So let's say if we only consider sexual reproduction and we want to find the limit of M for which the slope of the fitness with respect to time is positive, then we can differentiate it and we get this condition. So M should be less than eta times root of f into 1 minus f by g. So note that M is less than 1 by root of g into some constant. And for sexual reproduction, the same thing boils down to 1 by g into 1 by 2 times delta f square. So do note that here the dependence is on root of 1 by under root of g, and in asexual reproduction the dependence is on 1 by root of g. So the maximum tolerable mutation rate is much higher in sexual reproduction. It is higher in the order of root g times greater than, it's root g times the one that we found in asexual reproduction. So the longer the length of the genome, the sexual reproduction would do better. Now this also implies that if in case of asexual reproduction, if there are m into g flips, average of m into g flips, then the probability that no bits are flipped is e power minus mg. And for asexual reproducing species to have an individual to have an offspring that is identical to it is e power mg, assuming that mg is greater than 1. So for a species to persist, it needs to have an exponentially huge number of offsprings, or rather favorable mutations need to be very high and so on. So the clonal offspring is very rare in some sense compared to the sexual reproduction part. Yes, so now I'll hand it over to Saurabh, so he will explain some of the information theoretic aspects of this model.
Yeah, thank you, Burhan. Now we'll start with some information theory aspects of this. It's not very fancy information theory, so there's no rate distortion theory or even stuff like mutual information. We just want to make a connection of information entropy and then move on to the thermodynamic side of it. Look at Maxwell, Riemann, etc., and understand what is happening here. Why is evolution taking place, etc., from a thermodynamics dynamics / information perspective. So I'll just introduce the definition for entropy for those who might not know this. So if you have a random variable x with n probable outcomes, each outcome has a probability pi, then the entropy is given by minus sum of pi log pi to the base 2. If the base is 2, then you have entropy in bits. If it's e, then it's nats or something. Yeah. Events are equally probable. The entropy is simply log n to the base 2. This is the number of bits required to specify what has happened. So you could also interpret this in terms of binary search, since we know exactly about what are the events that can happen. So we could, in principle, arrange them in some ascending descending order and then perform a binary search, since it's the most efficient algorithm. And binary search is basically yes no question. So yeah, it gives you the number of bits. And if all PIs are not equal, then we will not use something by a binary search. We'll modify it. And you could derive, like, it's not necessary for this to derive, but as a side note, I would like to give some intuition of why this happens. Everyone knows this is pretty intuitive, but why pi to the pi log pi? I just want to motivate this in short before we move on to biology. So to derive this, assume n events again, each with probability pi. Construct blocks of length m into pi. The total number of blocks is m because pi is sum to 1. To determine one element in this entire block, you would have to ask log m questions. But in this whole block, there are sub blocks where we don't care. We just want to ask what event has happened rather than what point we are falling into this block of length m. So we would subtract those questions. So we fall inside a block I with probability pi. So we subtract those things. So the number of expected number of questions one would ask to determine if you what event has happened is log m minus this thing, because we are doing some extra counting here, and this gives this. No need to know this or something. It's not needed in the lecture, but I just thing I would like to motivate, because how is this the number of yes no questions is not clear to everyone when they first encounter this expression for entropy. Entropy is a measure of uncertainty of system. Information is gained when uncertainty decreases. So information is gained when entropy decreases. After an outcome of probability p occurs, we gain log 1 by p bits of information. And again, yeah, so if a pair coin happens, then after we get heads, we gain 1 bit of information. Before we didn't know what could happen. That 1 bit could be 0 or 1, but afterwards we exactly know. And if an event is very likely to happen with probability 1, after that happening, we have not gained much information. So this is what this thing, this form comes from. Of course, the derivation for this implies other things like independent events probability is multiplied, but information should add, etc. Yeah, but we'll just use this form. And entropy is the weighted sum of these. So this is the information gained after one event happens, but that event happens with probability p. So you add all of these up, and it is the information required to specify the outcome of each variable. This is the information we gain after that that event has happened. So now we go on to the biology part. We again move on to the model. Yeah. So if the normalized fitness is 1, that means each of the organism has figured out which of the two bits are better suited for survival. So we have G bits of information because G is the size of the genome. And here for our definition, we just say that if the normalized fitness is half, then basically you can just obtain that by simple random mutations. So the organism has not acquired anything about the environment. We assume ergodicity / homogeneity assumption for obtaining this expression, that each of the bits f has a probability fg, but fg is equal for all g belonging to capital G. So fg is equal to f for all g belonging to capital G. So we define the information acquired with respect to the fully random case, which is each of the bit has probability half, to be this in accordance with our definition for information here, which is log 1 to the base b, and this is simply log 2 minus log 1 over fg. This is the expression for information acquired when the population has a mean fitness of small f. Plugging in the homogeneity / ergodicity, we have fg equal to f for all g belonging to capital G, capital I, and then you just sum this up. The a term inside the summation no longer depends on small g, so you get this expression for information acquired when the normalized fitness is small f, or small f is capital F by G, so we just substitute this over here. And we differentiate this with time to get rate of change of acquired information is equal to capital G times rate of change of fitness upon the fitness. So it's fractional rate of change of fitness. Note that in this model, we have not really assumed any model. So there could be something like dominant recessive genes, or the summation, the linear model, which we had fitness equal to sum of xg, that may not hold. This is just if the population has f, so it's really very model independent. There are some caveats, but yeah, it does not depend if it's sexual reproduction or sexual reproduction with mutations, or if there's some other non-linear definition for fitness or something like that. This is just the expression, the general expression for information acquisition with time, relating it to the rate of change of fitness with time. Yeah, so if small f is equal to 0.5, roughly the rate of information acquisition is twice the rate of change of fitness into G, of course, capital G. And we derived small f of t for sexual and asexual reproduction. Capital F is just G times that. And we observed that small f of t, rate of change of small f of t, was bigger for sexual reproduction rather than asexual reproduction. In terms of information, this simply means that sexually reproducing species acquire root of G bits per generation, while asexually reproducing species acquire 1 bit per generation. How can you explain the faster increase in fitness in sexual reproduction? Because after you select the organism not only knows that it belongs to the top 50 percentile, but when it participates in the next stage of mating, it also gets a chance to interact with the other organisms and acquire more information about what the overall distribution for the fitness is for the whole population. In asexual reproduction, the organism basically mutates on its own. It does not depend on what the other organisms are doing or something like that. So in sexual reproduction, you can say that you acquire more information per generation. So that is why from this expression, you get a faster increase of fitness in sexual reproduction. This observation that fitness and increase in information acquisition increase accompany each other is very important. We'll come to that after we take a slight detour to Maxwell's demon. But keep this in mind. Also keep in mind that the expression we have here is pretty much model independent. So we'll keep these 2 points in mind. We'll go to Maxwell's demon and then conclude all of this, when can evolution happen, etc., from an information perspective. So the Maxwell thought demon experiment is basically this. You have a container of ideal gas, and you have so the door basically measures if there's a hot particle coming near the the demon basically measures if there's a hot particle coming near the door. It lets in to the other side, and it rejects a cold particle, and it lets a cold particle moving from the other side into the left side, etc. And so with time, you expect that you will reach a separated state where the right side has hot moving particles and left side has cold moving particles. This seems to defy intuition. Can you construct a perpetual motion machine by this? This is a very old problem. Maxwell formulated it around 1870, and it took about 90 years to get a proper resolution of this. Everyone knew that there was something fishy about this, violating second law of thermodynamics, but solution to this was arrived by Landauer, who was a research scientist at IBM, by the following argument. After getting a result of a measurement, the measurement can be reversible. So there's nothing like measurement being irreversible is a clue to not a violation of second law of thermodynamics. Measurement can be reversible, but even that, Tiemann has to erase some information he writes in his scratchpad about the velocity of the particle, and then some bit going from a very certain state to a random state is increase in entropy. So that increase in entropy is at least greater than or equal to this decrease in entropy when you separate this ideal gas into chambers of cold ideal gas and hot ideal gas. Even if the demon does not erase it straight away, Bennett of the quantum communication fame DB-N84 protocol showed that however well prepared the demon might be, he will eventually run out of storage and erase information. Erasing randomness is very hard. Resetting a n-bit resistor requires n-kT ln to amount of heat. Are you saying that when I erase randomness on my computer, that is, I reset a unknown register to some fixed state, my computer is dissipating heat? Yes. Modern transistor-based computers dissipate around 500 kT ln to the base heat. Ln to heat. This is pretty bad if you see compared to this amount kT ln 2. For 1 bit, it would be kT ln 2. This is 500 kT ln 2 per bit of dissipation. A reversible computer would dissipate kT ln 2 of heat. Again, this is a small digression to thermodynamics and computation. But of course, our reversible computer example, quantum computer would do this task by dissipating the minimum amount of heat possible because the computation is reversible. But of course, your computers are more reliable. So the very fact that you plug it to the power socket and dissipate so much heat means that you get reliability. This large expression also points to error correction. You may not have error correction implemented by coding or something. It may simply happen by the coordinated movements of electrons in a transistor or something. Yeah. So then the entropy of no isolated system can decrease with time. Relating information and entropy in inverse form kind of thing. We can say that no isolated system can gain information with time. So coming back to our model, we saw that fitness was increasing definitely. And by this equation, we have that information and information acquisition increase is linked to fitness increase. So what is happening? Why are we getting evolution? What does this mean from an evolution, from an information perspective? So what is really happening here is that we selected the fittest 50 percentile to survive to take part in the next stage. The bottom half, you can assume, died out and the bits became more random. So the entropy gained by the bottom half, the bits becoming more random, is at least greater than or equal to the decrease in entropy of the selected population. Of course, you need not be so cruel. Death is not so necessary. You could assume that the environment is a Maxwell's demon that select the fitter population half and separates them. And then the increase in entropy is by the erasure of information in the Maxwell demon scratchpad or something. So the conclusion is pretty striking as well as very intuitive. I mean, humans have been knowing this subconsciously for thousands of years. We have selected crops and all. That if we had not separated the unfit population, these would have bred with the fit population, etc. with time, and we would not have gotten any increase in average fitness. Removal of these from breeding process is necessary for evolution because we want to lose some information of some subset to get an information increase in the other subset we are interested in. to get fitness increase. So some form of information erasure in other subset is necessary for fitness increase. You could do that by killing basically half of the population, separating it by or some other process. But yeah, information acquisition and fitness increase go in hand. Yeah, so that is basically the picture of this fitness increase from an information perspective. I'll hand over the presentation to Burhan.
So thank you, Saurabh. Now we will dive into the biology part of it. So now it is very counterintuitive. Why would a population have males in the first place? Because males, they don't really reproduce on their own. They have they consume the resources almost equal to what a female would consume, or female, or a parthenogenetic female. Parthenogenetic female is the one which can reproduce on its own without the need of a male, so it's basically adding to the pressure of the species. So here Mackay proposes this model in which we consider 50% of the population is parthenogenetic female, and other 50% is a sexually reproducing couples. So and both of them they produce same number of offsprings. And what makes an individual parthenogenetic female is basically one of the bits which we can call it as a parthenogenetic gene in some sense. So if that gene is present, the female can reproduce on her own without the need of a male. So here we have two models. So where we have mg equal to 4, so basically the rate of mutation is much higher. And here we have mg equal to 1, so the rate of mutation is less. So on the x-axis we have fitness, and on the x-axis we have time of evolution, and here we have fitness. Here we have percentage of parthenogenetic mothers or females, the ones that can reproduce on its own. So the dots are the fitness of the parthenogenetic individuals, and the continuous line is the fitness, sexual fitness, or the ones that require male and the female. The parthenogenetic female would not have much diversity. It would be very much like asexual reproduction. One biological organism that you can think of is C. elegans. It's a worm. So there are two kinds of sexes in C. elegans. First one being the hermaphrodite, which is basically a parthenogenetic female, and there are males. Okay. So now coming to the model, we start with around 66% of which are parthenogenetic females, and then the simulation is run. Now, as Saurabh showed earlier, that the sexual reproduction leads to offsprings having higher tolerance of mutation rates. Because of that, a sexually reproducing organism can scan through the different possibilities much faster than a parthenogenetically reproducing female. So that's why we see that that as soon as the population is evolved over time, most of the parthenogenetically reproducing females die out. Of course, there are some clusters, there are some pockets in which these are found. However, the sexually reproducing organisms, they sort of thrive in this sort of environment in both the cases. Then once a good fraction of so sometime around this point, when a good fraction of the individuals have reached the optimal fitness or the maximum fitness, the parthenogenetic females kind of take over. Why this happens? Because the explorability is less in a parthenogenetic female. So it's more like clonal offsprings rather than a combination of two. So that's why these are not so sexually reproducing organisms are non-clonal, and then there will be variations because of which there is a chance that they fall out of the optimal, which we see here. And because of this, the parthenogenetic female takes over almost the entire population after reaching a steady after reaching the maximal fitness. In the case where the the mutation rate is low, but in the case where the mutation rate is high, it is observed that parthenogenetic female never takes over. So, because in some sense the fitness function, you can say that it changes really fast. So, and this observation from our models, so some of these implications of the model are consistent with the argument or the hypothesis proposed by Haldane and Hamilton of kin selection. So kin selection basically says that a kin or a group that are highly coordinating, they have a much higher chance of survival rather than the other communities. So so in an unstable environment where the mutation rates are high, or rather not mutation rates, but where the fitness function changes or the real fitness function changes, there the sexually reproducing organisms thrive. So here I would like to draw in the biology. Most of the multicellular large organisms, they reproduce sexually, and it makes sense why they have high mutation rates, or rather high. I mean, they don't really have high mutation rates, but they have very frequently changing survival function. So one example is the one that I showed you earlier in the case of malaria. The parasites evolve really fast, and these parasites sort of change the fitness function really fast. So in that sense, it makes sense to have sexually reproducing communities where you have the whole population having a copy of a fit gene or something that of a fit gene, yes. So that sort of so our model is kind of consistent with this hypothesis of kin selection. Now the second assumption that we had is the additive fitness function. So we just took the sum of all the individual XGs or the genes or the nucleotides without really and we just assume independence. So in some sense we took sum of independent terms. So is it a reasonable fitness function? Now, according to Maynard Smith, the more good genes you have, higher you come in the pecking order. So, if you have more good genes, you are evolutionarily quote unquote fit. The more fit you are, and then there are a fitness function based on this particular argument that are extensively used in population genetics, and it is consistent with some of the so there is some empirical evidence that this is a good enough fitness function. But some of the implications that I would like to highlight is that real fitness function might involve multiple interactions of individual genes. So let's say, for example, height, we don't have a gene that encodes for height, but it's a set of genes, different genes interacting with each other, and they give rise to different heights. And of course, there is this environmental factor also, but also the genetic part. In that case, the crossover or sexual reproduction might actually reduce the average fitness of the individual. But we saw from the model that it is consistent with what we observe in nature. So from our model, we can predict that evolution would have favored species that use a representation of a genome. So here we don't really know the representation on how can we represent the genome, but a representation of the genome that corresponds to the fitness function that has only weak interactions, because we want independent terms. And for independent terms, we would to satisfy that, we would want to have weaker interactions among these different elements of the genome. Or at least it should be, even if there are interactions, it should be such that it is a sum total of these individual elements. So this has been used by several modelers and systems biologists who are working on this. And with this, we would like to conclude our talk. So thank you, and we would be happy to take questions.
Thank you, Saurabh and Burhan, for this really nice talk. So one of the questions was that what inspired you to work in this interdisciplinary area? Like one of you being from EP and one from bioengineering?
No, I was just reading David McKay's book as a pastime. I could not take information theory due to a slot clash with another course. So I've been reading it during the semester, and then I just came across this chapter. The math seemed okay, but of course there were many assumptions which I don't know the real life. I'm just pretty okay at math, but I don't know how what assumptions we have taken are they good enough or not. So I took help of Burhan, who did a brilliant job today. So
okay. It was a very nice.
Yeah, go on.
Oh, sorry, continue.
Okay, so for me, I was working on a project that required me to learn some information theory, and I also came across David McKay's book, and me and Saurabh were also talking about the book, and that's how we came up with this.
That's great. Like, it's nice to see such interdisciplinary research happening even at an undergraduate level.